Hello, I'm Daniel Bright, and this is my live stream for the Bio 375 project for lesson 1. I'm doing this by myself, so let's get started. So, question 1. Suppose you perform the following test cross, and then it gives two individuals with a specific genotype. Then assume that all of these genes undergo independent assortment. The part is, what proportion of the offspring is expected to have that genotype? So to answer that question, I made a monohybrid cross for each trait A, B, C, D, and E, and I took the proportion of gametes that would have each trait. So, like for example, the first trait is big A, little a, and out of the 4 combinations for the A trait, only 2 of 4 will be big A, little a. So that's 1 out of 2. And I did the same thing for the B trait, C, D, and E. And the answer I got was 1 out of 128. I just simply took each fraction for each monohybrid cross and multiplied them by each other to get 1 out of 128. I did the same thing again for B and C. For B and D. Now for C, one of the traits is little d, but the thing is, there is no combination for the D trait that is little d, little d. They each have at least one big D in them, so that doesn't work. So the answer to C is none will have this genotype. Okay. So question 2 is fill in the blanks. Part A, paired homologous chromosomes are sometimes called blank or blank. Okay, so when homologous chromosomes are paired up, they come together, they're called sometimes bivalents because they have 2 homologous chromosomes coming together, or they're called tetrads because each chromosome is made up of 2 bivalent. Each chromosome will be made up of 2 sister chromatids. So there's 4 sister chromatids coming together. So it's a tetrad. So it could be called a bivalent or a tetrad. B. The structure that connects a centromere to the microtubules of the meiotic spindle is called I believe that is, hang on one second, I'm sorry. It's called a kinetochore microtubule. Okay. And then C. A diploid fruit fly has 4 pairs of chromosomes. Okay, so if there's 4 pairs, it means there's 4 chromosomes here, and they have their homologous counterparts. That's 8 chromosomes in total. A fruit fly cell in prophase 1 has blank sister chromatids. Okay, so in prophase 1, the cell is not split yet. It's still one cell, which means there are still 8 chromosomes there. And for each chromosome, there are 2 sister chromatids. So if there's 8 chromosomes, then you just multiply 8 by 2 and you get 16. So at this stage of prophase 1, there are 16 sister chromatids. And then, while a fruit fly cell is in metaphase 2, it has blank sister chromatids. Okay, so in metaphase 2, the cell is already split, which means if the 8 chromosomes has split, so there's 4 in this cell and 4 in this cell. And again, for each chromosome, there's 2 sister chromatids. So in this stage, 4 chromosomes each have 2 sister chromatids, which means there are 8 sister chromatids in this phase. Okay. Moving on. A blank is a type of genetic cross that can determine if a purple flower pea plant has the big P, or big P, little p genotype. That is called a test cross, which is where you cross two individuals to see, just to test to see what happens. And E, a non-functional allele is sometimes called I believe that's a null allele. Yeah. The answer is null allele. Null means like none, not non-effective. Yeah. And then, while a partially functional allele is called a hypomorphic allele. F. Mendel's law of Blank involves two traits. So that would be independent assortment. It means the way how, like for example, with the pea plants that Gregor Mendel tested, the trait for tall, or the trait for plant height, the trait for plant height assorts independently of the trait for something else, like say flower color, for example. They sort independently of each other. Just because a plant is purple, that does not mean it's going to be tall. It could be purple and short, for example. This isn't true for all. Pardon me. This isn't true for all traits. Some traits do not assort independently, but some do, especially the ones that Gregor Mendel was testing with his pea plants. Well, Mendel's law of Blank involves a single trait, because that's segregation. So what you got is, you have two chromosomes, and each one has a different allele for the same gene on the same locus. Those two alleles will separate from each other and go into gametes individually. So, which means about half. Yes, about half of that individual's gametes, not about half, it is exactly half. Half of that individual's gametes will have this allele, and the other half will have this allele. Okay. Okay, moving on. Okay, so question 3. Suppose an organism has the genotype, and it gives a genotype. Assume that the alleles for the genotype given assort independently. Okay, so 3A, diagram all possible homologous chromosome arrangements in this organism during metaphase 1. Be sure to indicate which alleles are found on each pair of homologous chromosomes. Okay, so the way it works is, in metaphase 1, there's the chromosomes align along the metaphase plate. It's an imaginary axis in the middle of the cell. And so you'll have chromosomes up here, and their homologous counterparts will be underneath the metaphase plate. And you could have all paternal up here and all maternal down here, or some maternal up here and some maternal down here, and just all kinds of combinations in between. And I illustrated that in another image. And part B, indicate all of the possible gamete genotypes that can be produced in this organism at the conclusion of meiosis II. Okay, so to do this, I used a branching fork line, I think it's called. Hang on one second. Yeah. Sorry, it's called a forked line method. I used the forked line method to determine what the genotypes will be at the end for all the traits. And okay, so the answers are big T, big R, big Y, big T, big R, little y, big T, little r, big Y, big T, little r, little y. And there's no little t because the organism only has big T, which means all its gametes will have big T, no little t's in this one. All right, so hang on, having some trouble here. Okay, here we go. Next question. Question 3, part C. How many sister chromatids are found per cell in this organism during metaphase 1? Okay, so this is where we need to clarify something. So if this organism if the alleles assort independently, and if there are 3 traits for T, R, and Y, that would mean this organism will need to have at least its chromosome set would need to be at least 3 big, because each trait, in order to assort independently, would have to be on a different chromosome to ensure that they always assort each other independently of the other 2 traits. So there's 3 chromosomes, and they had their 3 homologous pairs. At least 3. We'll just assume for now that it's just 3, just to make it simpler. So part C was, how many sister chromatids are found per cell in this organism during metaphase 1? So hang on one second. Okay, sorry about that. So you have 3 chromosomes here, you have their 3 counterparts here for 6 chromosomes. And for each chromosome, there are 2 sister chromatids, which means that there are 12 sister chromatids during metaphase 1. And part D, how many chromosomes are found per cell in this organism during metaphase 2? So in metaphase 2, the cell is already split. There's 3 chromosomes in one cell and 3 chromosomes in another cell. And each chromosome still has 2 sister chromatids, which means in metaphase 2, there would be, we're not working with chromatids, we're just working with chromosomes. So in metaphase 2, there's 3 chromosomes, and that's it. That's the answer. 3 chromosomes in metaphase 2. All right, question 4, part A. A horse has 32 pairs of chromosomes. Okay. Part A. How many chromosomes are present per horse cell at the conclusion of meiosis? Okay. So at the conclusion of meiosis 1, the cell is already split. Okay. Now, with that in mind, since there's 32 pairs of chromosomes, that would mean that 32 chromosomes went here and 32 chromosomes went here into each cell. So the answer to part A would be 32. 32 chromosomes per cell at the end of meiosis 1. Part B. How many tetrads are present per horse cell during during prophase 1 of meiosis? Excuse me. So a tetrad, as I said already, is where you have 2 homologous chromosomes, which are made up of 2 sister chromatids joining together, right? So, and since it's 2 chromosomes coming together, it means there's 1 tetrad for every pair of chromosomes. And since this organism has 32 pairs of chromosomes, then there are 32 tetrads during prophase 1 of meiosis. And part C. How many sister chromatids are present in a horse cell during prophase 1 of meiosis? Okay, so there's 32 pairs of chromosomes, and for each pair there's 2 chromosomes, which means there are 64 chromosomes. And for each chromosome, there's 2 sister chromatids. And 64 times 2 is 128. So there's 128 sister chromatids during prophase 1 of meiosis. Because in prophase 1, the cell is not split yet, it's still one cell. And part D, how many sister chromatids are present per horse cell during metaphase 2 of meiosis? Okay, so in metaphase 2, the cell is already split. So there's 32 chromosomes here and 32 chromosomes here. And for each chromosome, there's 2 sister chromatids, which means there's 64 sister chromatids during metaphase 2 of meiosis. Last part. E. How many bivalents are present per horse cell during anaphase 2 of meiosis? So in order to have a tetrad, you must have a chromosome and its homologous counterpart. But those two are split during anaphase 1 of meiosis, which means in anaphase 2 of meiosis, they're already long gone. So there are no bivalents per cell during anaphase 2 of meiosis. Okay. Question 5. Consider the following tetrahybrid cross in pea plants. And then it gives the genotype of 2 pea plants. Assume that, and then it gives the traits that each gene governs, and which one is dominant, which one is recessive. Assume that the alleles of all 4 genes assort independently. Okay. Question 5, Part A. What proportion of the offspring will be tall, round, green, and terminal? Okay, so giving us a specific phenotype. Okay, here we go. So with phenotype, since we have dominant and recessive alleles, it means that if the dominant allele is in any combination of gametes, the dominant allele will express itself and the recessive allele will not express itself at all. So in tall, round, and wait 1 second. Yeah, pardon me. So tall and round are both dominant traits. Green and terminal are both recessive traits. And so, one moment, please. Okay, so since tall is dominant, like I said, anytime you have big T, it's going to portray itself. So what I did was I did a monohybrid cross for each trait, just like I did before in question 1, I think it was. And for T, you have 3 out of 4 that have the big T allele. So it's 3/4 for T. Then for round, again, round is dominant, and 3 out of 4 will have the big R allele. So that's also 3/4. The next one is green. Since there's only one organism that has a Y allele, since it only has one Y allele, or rather big Y allele, it means that 3 of the 4 individuals produced will be green. So it's also 3/4. And the last one is terminal. Now with terminal, I just realized something. Oh wow, I just realized I made a big boo-boo. So wrong again. It turns out that the trait for color, yellow versus green, 2 of the 4 will turn out to be green. 2 of the 4 will be recessive, so it's half. half will be green, and half will be axial instead of again. Okay, so yeah. Okay, so yeah. So for tall, 3 of 4 will have the tall allele. Round allele. For excuse me. For color, 2 will have the green. Yeah, 2 will have the green phenotype because only 2 will be dominant recessive. Only 2 of the 4 will be axial. So it's 3/4 times 3/4 times half, which is 9 out of 64. Okay. Then part B. Let me pull up the question again. Part B. What proportion of the offspring plant has, and it gives a specific genotype. So now we're looking for genotype. Okay. So for little t and little t, for big R, little r, it's half because only 2 of the 4 will be will be heterozygous. Okay. Then we have big Y and little y. Okay. So okay. Yeah. For what was the last trait? Big A, little a. Okay, so that is also heterozygous. And that would be half again. So it's 1/4 times half times half times half. Which is question 5, part C. What proportion of the offspring will be heterozygous for all 4 genes? Okay, now we're just looking to see which ones are heterozygous. So for T, it's 1 out of 2. For R, it's 1 out of 2. For Y, it's 1 out of 2. And A. Okay, so for all 4 of them, for all 4 traits, the likelihood of the individual being heterozygous for that trait is 1 out of 2. So when you multiply them all by each other, you get 1 out of 16. There's a 1 out of 16 chance the individual will be heterozygous for all 4 traits. Then finally, part D. Part D was, okay, what proportion of the offspring will be dwarf, wrinkled, yellow, and axial? So again, we're looking at phenotypes, not genotypes. So for dwarf, it's 1 out of 4 because dwarf is recessive. So the recessive trait will only express itself if it's the only one there. So 1 out of 4 for dwarf. Okay. The next one was wrinkled, which again is recessive. So it's 1 out of 4 again. Okay. And then next one is yellow. So for yellow, since yellow is dominant, it means half because you only have to have one big Y for in order for the dominant trait to express itself, it only needs to have one allele. So half will be yellow. Okay. Sorry about that. Okay, the last one, last trait was axial, and axial is recessive, and only two of the four will be axial. So it's 1/4 times 1/4 times half, which is 1 out of 64. Okay, so there you go. Those are my answers to the questions. Thank you all so much for watching.